Okay, dear students, welcome for the lectures of physiology, lecture number 15. We will see or continue the excretion that we have seen previously, how the nephron is working, how the ultrafiltration takes place. Now, what is the secretion or secretory activity carried out in excretion, and what is the counter current multiplier exchange theory? This is very important topic that we will see here in details. In secretion or tubular secretion, particularly in mammalian kidney, the urine formation is carried out by the tubular secretion, and this is one of the most important process which requires energy, or we can say this is the active method. Mammals produce hypertonic urine, so urine have different concentrations, low, medium or high, and accordingly the name is given to it, that is produce hypertonic urine. And the phenol red and para amino hippuric acid is the chemical that filtered with urine or secreted in the tubular secretion, generally known as PAH, are secreted by the renal tubules in the tubular secretion. And some of the foreign substances which are toxic to the body are also eliminated by tubular secretion. Example, some of the phenolic compounds and their products of detoxification. Actually, the nitrogenous waste is also a toxic material in which the body eliminates unnecessary or some harmful chemicals or materials thrown outside by excretion. So, in next topic is counter current multiplier exchange theory. In this method of excretion, or we can say reabsorption or ultrafiltration, all these activities are serially continue there too in the kidney. But in counter current multiplier exchange theory, specific ions which are important and necessary for the human body or physiology are, if accidentally removed or cloned or filtered, that are definitely reabsorbed, or they maintain the pressure, concentration, osmotic pressure, everything accordingly as per the weight of the tissue or body or human being or animal, we can say, and that is nothing but the counter current multiplier theory. So the loop of Henle play a key role in counter current multiplier exchange theory, or we can say counter current multiplier exchange activity, because the fluids in the descending and ascending limbs of this loop is in opposite direction, that is counter current flow, and therefore this is known as counter current multiplier theory. So one limb is descending limb and another limb is ascending. We have previously studied in the last time in the diagram the concentration of fluid in between both these loops is different, and which additive and shows multiplication effect is, so the urine system is known as counter current multiplier system, or we can say counter current multiplier system. So you can mention here or bold it in another color so that you will read it. Counter current means it counts the material, it counts the pressure, it counts the concentration in both loops and tries to maintain the balance in between these two by changing the ions, and this is the counter current system. The length of loop plays a key role in final concentration of the fluid. The loop of Henle is deeply rooted in medulla because the half part is in cortex and half part is in medulla. So loop of Henle is definitely in medulla region or lower region. The concentration of solutes increases in descending limb of the loop because sodium ions are actively passes into the interstitial fluid, and sodium is the main element in the material. This produces osmotic gradient in medulla. So this is the important way. This is the important method through which the excretion is specifically carried out with the pressure, or with the maintaining the gradient, osmotic gradient. And this is in the medulla region, causes the passive diffusion. Active diffusion needs energy, but passive diffusion is a simple, of which sodium into descending limbs. Simultaneously diffusion of water also takes place, and the fluid leaving the loop, or the fluid which comes out of loop of Henle, has osmolarity about 100 milliosmolarity per kilogram, and the osmotic concentration of the fluid which is there secreted at the tip of a renal papillae can become high, and it may be up to 1200 to 1400 milliosmolarity per kilogram, and this is the way through which we can calculate the osmolarity. At last, finally, the fluid reaches to the distal convoluted tubule because of hypotonic nature, which is regulated by the antidiuretic hormone, ADS hormone, and the collecting duct progressively becomes hypertonic because the diffusion of water and finally formation of urine, or we can say urine is formed. In this way, the actual ultrafiltration, by maintaining the osmotic gradient, by maintaining the ion balance count, and this is a whole procedure taken place particularly in the loop of Henle, is called as counter current multiplier exchange theory. So if we will see in detail how the counter current system is working, we will see in diagram. This is the counter current mechanism seen between the nephron tubes and the collecting tubule. This one is the collecting tubule. In collecting tubule, the H2O is get absorbed, urea is reabsorbed, we can say, and the osmolarity pressure is about 100 to 1300, or 1300. Here 1400. So this is the difference in between this collecting tubule and a loop of Henle. This is the glomerulus. Here the ultrafiltration takes place. This is afferent tubule. This is efferent tubule. One is bringing the blood, another is carrying the blood. In between, the glomerulus filters the urine, nitrogenous waste, materials, ions, solute, everything in the proximal tubule. And this is the distal tubule. Next part, and this is loop of Henle, where the exact counter current multiplier exchange theory or exchange system is seen. In this tubule, both the tubules shows different pressures, both the tubules shows different concentrations. Here is the 400 sodium chloride concentration, Na plus is here, or NaCl sodium chloride, because the main part of urine is salt, or we can say NaCl. Therefore, sodium and chloride ions are always there in urine, and they maintain the osmolarity as per the need of the cell, body, or system. So here the active transport is being carried out, and this pressure of 300 osmolarity is down, slows down here up to 200, and to regulate it, to maintain, the water is percolating outside the tubule and urea is entering here, and here urea in both way entering and removing, and sodium chloride is also removed from the tubule. So sodium and chloride, these are the molecules. So in this way, this pressure from 300 to 200, it raises up to 1400 and again slows down up to 150, and then the salt material is filtered here. Here the juxtaglomerular, I have told you previously that the complex where the reabsorption takes place in different parts, where artery and vein, both are here, and exchange is here. This is the cortex region of this belt is the cortex belt, and this is the lower part of the kidney, that is medulla, or inner part of the kidney is medulla. So this is vasa recta, because in afferent and efferent tubule, the vasa recta is also a tube which maintains the gradient of the concentration or ionic balance of the waste material. So you see 100 to 500 and here 100 to 1300. This is the great difference that the collecting tubule shows and vasa recta shows, as well as in the loop of inlet. This is the very quite different changes, 300 to 1300, here 300 to 1400, and here 100 to 1300. So this is the great changes occur in the osmolarity pressure and excretion. So this is the osmolarity exchange, or we can say counter current multiplier theory or counter current multiplier system in kidney. Now, what is the composition of normal urine? Normally, in human urine, salts are then yellowish bile pigments, which gives color. Actually, molecules or materials are not filtered, only the color is given back. So, normal human range of urine production is about 0.6 to 2.5 liter per day. About 96% of urine is composed of water, and the remaining 4% contains organic and inorganic compounds with some hormones and various metabolites. They are also filtered from the body if not properly digested, absorbed, whatever. And urine also contains total solids, which are about 55 to 58 grams per person per day. The proportion of various organic and inorganic elements in mammalian urine varies person to person as per the age, health, and diet changes. Yellowish color pigment is derived from the products which are formed by the breakdown of old red blood cells. In human body, RBC have specific life, and after that it broken down and form pieces. At that time the hemoglobin is dissolved in the water and urine. So color urine gives yellow color or yellow reddish color. So this is the normal composition of urine, in which the organic constituents and inorganic constituents are there. Inorganic constituents: urea, ammonia, uric acid, creatinine, and ascorbic acid is found, and these are 80 to 90% of the total. 2.5 to 4.5% is ammonia, 0.7 gram per day uric acid is formed, 1.2 to 1.7 gram per day creatinine is excreted, and ascorbic acid is about 15 to 50 milligram per day. So this is the composition of inorganic constituents which are released from the kidney in the form of urine in man, or we can say the urea composition and percentage. Inorganic constituent of urine contains chlorides, phosphates, minerals, nitrogen, oxygen, hydrogen. And among the minerals, so these are very important minerals, that is potassium, we can say, then sodium, calcium, and magnesium. So these are the elements, or these are the mineral groups which are necessary to maintain the osmolarity, or we can say excretory secretory product or secretory material or secretory pressure. So everything is maintained regarding to maintain the physiology. So chlorides are about 6 to 9 grams per day secreted in urine or contains in urine waste. Phosphates are 0.8 to 1.3 gram per day. Minerals sodium, potassium, calcium and magnesium are about 4 to 5 gram, potassium 2 to 5 gram, calcium 1.1 to 0.3 gram and magnesium 0.1 to 0.2 gram per day. This is calcium and magnesium are secreted in very low amount, whereas sodium and potassium are the very important and necessary elements which are filtered or get by the body through the diet mostly. So in this way these minerals also filtered that in the urine. Then nitrogen, oxygen and hydrogen, these are also, nitrogen and oxygen are about to same in percentage per liter, whereas hydrogen have 1.51 grams per liter. So very little amount of hydrogen is excreted. So this is about the constituents or composition of urine. If kidney is not working properly, or if physiology of kidney is failed to respond properly or normally, then we can call it as a kidney failure. So kidney failure means, or we can describe about or explain about when normal kidney becomes unable to filter adequate waste products. Adequate means very necessary to be filtered from the blood is the adequate waste product, and the medical condition of such a situation is known as kidney failure or renal failure. So there are two types of patients in kidney failure. One is acute and second is chronic, or we can say acute and chronic means short and long period of such a situation or results of kidney. In acute kidney or in acute kidney failure, proper treatment may reverse the proper or normal functioning, but in chronic treatment there are very less or no chances to recovery. Therefore, the acute treatment or acute situation is ever normal, or we can say ever good, because it can recover. But chronic treatment have low chances or no chances to recover the working of the kidney, or kidney failure we can say. So kidney failure may be determined by the decrease in glomerular filtration rate than that of its normal rate of filtration, and this can be determined by detecting the creatinine or urea content of the waste product. So creatinine or urea contents are the responsible factors for the rate of excretion of material. This is generally checked in the waste product or in the blood. In urine may be detected. There are two categories of kidney failure, acute and chronic, two types of kidney failures. The solution is dialysis or kidney transplant in the body of patient. This may be caused due to the excess use of the drugs. This solution generally maintained by the machine called as dialysis machine or kidney transplant machines, and this is very simple. So acute kidney failure is a small period, chronic kidney failure is a large period. So these are the two types of kidney failures we have already seen, but we will discuss something here about acute kidney failure. This is nothing but a speedy and progressive loss of adrenal functioning. The acute kidney failure causes sharp rise in nitrogenous compounds in the blood, and this may be caused by total collapse of fluid and electrolyte balance in the blood. whereas the glomerular filtration rate is very slow and very inadequate, means not properly or normally working at. In chronic kidney failure or long period failure is a long-term consequence in which kidney function is slows down and develops slowly. In chronic kidney failure, the kidney's basic function of ultrafiltration, or we can say glomerular filtration, is altered, means not totally working normally. Either low or high filtration rate is there. This can be studied by determining serum creatinine content. What the serum creatinine level is there, that decides the actual kidney failure situation, either acute or chronic. Such a kidney failure is irreversible in chronic. This is not reversible. Once happened, it will slows down and at last the kidney will fail. So the symptoms of kidney failure are weakness, shortness of breath, nocturnal urination, vomiting, diarrhea, blood in the urine, difficulty in urination, etc. These are the symptoms, or we can say the conditions which are created due to the kidney failure, or we can say not working properly. So this is about the acute and chronic kidney failure situation. How the artificial kidney is given in the kidney failure situation, that will be studied in the next lecture or next time. Till then, we will stop here. If you have any questions, you can communicate directly to my number or my message, or on WhatsApp number you can send the questions. So have a nice time. We will stop here today's lecture.